as because I am working alone, that's why I take this stand and the microcentrifuge tube with me. If there is other microbiologist or molecular biologist who are working in the lab, you can put the tube here in the box to transform from one end to another end. And as because this is the PCR extraction room, so you can transfer it through, and then they will open from that side. Now my first duty is to put this victim sample is on minus 20 degrees centigrade fridge. This is a minus 20 degrees centigrade fridge. I have to keep the victim sample here. Okay, now what do I need to do? This is the stand, and this is the ice pack. What I need to do first, before I proceed for the PCR extraction, I need to came in the vertical airflow room, where I need to mixing up the reagent. Okay, this is micro alicot stand. Okay, so right now my duty is to take the reagent for PCR extraction from the fridge. Now what we have, this is the Agapillium screen PCR reagent for SARS-CoV-2. This contains the Lium ampoule mixture tube, SARS-CoV mixture, SARS-CoV-2 mixture, which is prime, one positive control, one negative control. Okay, now what we need to do, we have to take the primer and put it in the primer section. Take negative control, put it in the negative section. Take the positive control and put it in the positive section. Now take the Lium ampoule 48 tube, which is already here. Now how we can take it. With the help of forceps we can take it, okay, and put it in the respective hole. But today we did not need 8 ampoule, that's why I have already make a cut. I already cut it and put it here, and carefully place this inside this seal pack so that it will be not contaminated. I already took four things, and this is primer, this is negative control, this is positive control. my first step is to put 15 microliter primer in each ampoule, in every ampoule. 15 microliter primer. Now you have to dispense the tip. Now cap the primer, put it in the primer section again. I already put primer in each vial. Now maybe you have a question that why I take the 4 vial, why I take the 4 ampoule vial. Now what we need to do, we have to put the negative control in the first and in the first alicot, or in the first ampoule we have to put the negative control, and in the last we have to put the positive control, and in between these two we have to put the sample for patient A and the sample for the patient B, means the RNA. Okay. Now what we need to do, we have to centrifuge the positive and negative control by putting it in the centrifuger. Now on it. After 2 minutes, close the centrifuge, open and take the positive and negative control. Now keep it aside and put the negative control in the negative section, positive control in the positive sections. Okay, now with the help of pipette, take 10 microliter positive negative control from the negative section. 10 microliter negative control, put it in the negative control section. Now dispense the test. Now close it. Now what we need to do, we have to cap the negative controls ampoule, properly capped. Until and unless it will be capped, it will be contaminated, so we have to cap it up. Okay. Now what you need to do, first you have to take 10 microliter sample from patient A and put it in the respective ampoule. Then take another 10 microliter sample for patient B, I mean the RNA, and put it in other tube. Okay, dispense it. Now close it properly. Need to close the microcentrifuge stick properly. Now close the alicot cap.